Hello to our friends joining us via recording. Today we are covering the last lab lesson. This lab lesson is about the special senses. There are 5 special senses. We are really just going to focus on 2 in our class today. We are focusing on the process of hearing and the process of sight or vision. So the models that we're going to ask you questions on are going to be ear models and eye models. Keep in mind that next week is a review week for you. We're giving you some extra time to make sure that you're prepared for the lab final exam. So we will have plenty of review sessions next week to prepare you for that lab final. But remember, there are a ton of Blackboard assignments and visible body assignments due for lab next week to help you review all the content to expect on that exam. So check that out. Lots of stuff to do next week, even though there's not a new lesson, to help you get ready for that exam. To help us dive into lab lesson number 12 about the special senses, we started with a group work activity to remind us of our cranial nerves. So when we talk about the cranial nerves, remember that there are 12 sets of cranial nerves. For the lab exam, we need to know their names, we need to know their numbers, what they do. So let's go through and relate several of these cranial nerves to special senses stuff, or the kinds of things we're going to talk about today. So let's start with one of our first special senses, the one we're going to spend a lot of time on at the beginning of class here, and that's the process of hearing, or the part of the body that collects auditory information. Can you help me out in the chat? What was the number or the name or both of our cranial nerve that collects auditory information, that does hearing? Yeah, so it's cranial nerve number 8. Cranial nerve number 8. You may see it in some places. Let me give you the Roman numerals for it. Cranial nerve number 8. The name of this nerve is called the Vestibulocochlear Nerve. The Vestibulocochlear Nerve, cranial nerve number 8. When we talk about this nerve, it has 2 parts to it. It has a part that comes from the vestibule of the ear, and a part that comes from the cochlea of the ear. Does anyone happen to know, is it the vestibule part or the cochlea part that helps us to hear? Do we happen to know? Where do we do hearing? Yeah, Kira's right. So the cochlea, if you've ever heard of those things called cochlear implants, when someone is deaf because their cochlea doesn't function, we can actually put in a cochlear implant to make that part of their ear work. So when we talk about the vestibulocochlear nerve, the cochlear part of this nerve helps us with hearing, with auditory information. Interestingly, the vestibulo part of this nerve that comes from a part of the ear called the vestibule, this is what actually helps us with balance, or our fancy word for it is equilibrium. So cranial nerve number 8 also helps us out with the process of balancing. Vestibulocochlear nerve, cranial nerve number 8. It helps you to hear and it helps you to balance. That's because it has 2 parts that go to 2 different places in your ear. When we talk about the process of vision, which cranial nerve helps us out when we're talking about seeing? What cranial nerve helps us to see? Yeah, the cranial nerve that helps us to see is cranial nerve number 2. Cranial nerve number 2 is called the optic nerve. The optic nerve has that crisscross or that X. It's actually a thing, we'll label it during our class today. It's something called the optic chiasm. Chiasm literally means X. So when we're trying to find cranial nerve number 2, this is the one that has a little crisscross X. We collect visual information with the optic nerve. We have several different eyeball muscles that help us to move our eyes so precisely. We don't really think about it, but your eyes are kind of constantly moving, and they're moving in very small amounts. So for us to be able to do such precise little movements, we need a lot of different information going to those muscles to help us with movement. So we have 3 different cranial nerves that we said helped us out with the process of eyeball movement. So my 3 cranial nerves that help me with movement, the first one is number 3, cranial nerve number 3. That one's called the oculomotor nerve, cranial nerve number 3. My next cranial nerve that helps me with eyeball movement is cranial nerve number 4. That's the trochlear nerve, the smallest nerve. What was the last cranial nerve that helped me with eyeball movement? What's that last one that helps me to move the eyeball? Yeah, exactly. Cranial nerve number 6. That's my last one that helps me out with eyeball movement. Remember, I had mentioned this when we did cranial nerves before. This is another wink, wink, nudge, nudge for us. Memorize these 3 numbers in a group. So 3, 4, and 6. If I know that these are in a group together, and if I know that at least one of them helps me with eyeball movement, that'll help me remember that they all help me with eyeball movement. So these each go to different muscles that are on the outside of the eyeball. We'll look at those different eyeball muscles during class today. The other special sense that didn't make my list here, we mentioned this before we went into groups, there is also the special sense of smell. Special sense of smell. I know I didn't ask you about this one, but which cranial nerve helps you to smell? Yeah, cranial nerve number 1 is a nerve called the olfactory nerve, the olfactory nerve. The process of smell is technically called olfaction. Olfaction, the process that you use to smell. So cranial nerve number 1, the olfactory nerve, helps you to smell. There are multiple cranial nerves involved in taste, so make sure to refer back to your packet on that. The only ones we're going to work on today, though, of our special senses, we're going to focus on hearing, focus on seeing, and focus on balancing primarily today. That brings us back to this pretty color-coded brain here with those cranial nerves. Remember, we must be able to identify them. So you've got that picture in your lab packet. You've got the review assignment to help you out with reviewing. Yeah, that's true. Kira mentioned too on this one, we can actually see some nice parts here in our spinal cord as well. That's true. So we can use this picture to review all kinds of things, right? Let's work our way through the cranial nerves that we listed before to find them. So remind me in the chat, when we talked about hearing, what was the number of the cranial nerve that helped us out with hearing? Or you can give me the name if you're really talented, but that's a long name. Yeah, cranial nerve number 8. Let's type that up here. Number 8, my vestibulocochlear nerve. Vestibulocochlear nerve, cranial nerve number 8. So what we'll do, actually, I should have done this to begin with, is we're going to go through and number our cranial nerves. Remember, with the cranial nerves, you always start in the front. And when you get to areas where there's lots kind of next to each other, you always start in the middle. So cranial nerve number 1 up here, that's my olfactory nerve. That's the one, remember, that helps us with smell, olfaction. We're not going to focus a lot on that today. Right behind it, you can see the cranial nerve. Right now, we're only kind of seeing half of the crisscross. But here's where our little optic chiasm would be, right here, cranial nerve number 2. Working our way down, then we get here to cranial nerve number 3. Cranial nerve number 3 is the oculomotor nerve. Can you remind me in the chat, what does cranial nerve number 3 do? What's its primary function? Yeah, so the primary function on number 3, its name is oculomotor. Literally, that means eye movement. Oculomotor, literally. So cranial nerve number 3 helps me with eye movement. Remember that cranial nerve number 4, which is my little pink one right there. Cranial nerve number 4 also helps with eyeball movement. And cranial nerve number 6, down here in the middle of the medulla, cranial nerve number 6. This big one right here is cranial nerve number 5. Remember that cranial nerve number 5 is called the trigeminal nerve, and it gets its name because it has 3 branches. Trigeminal, 3 parts. This was the one, by the way, if you stuck up your 3 fingers in the middle of your hand and you put them kind of at the corner of your mandible, that shows you where each of these little branches go. So it goes to all kinds of different places in your face. Cranial nerve number 5 right here. Number 6 was down in the middle. Then I've got this little green one right here that was number 7. Number 7 is another one that goes to the face. It's called the facial nerve. Next to it is the nerve that started this whole conversation, right? Cranial nerve number 8. So cranial nerve number 8 is this pink one that we see down here, cranial nerve number 8. This one, remember, is hearing and balance. So it helps us with hearing and balance with its 2 different parts. Next to number 8, then, we've got number 9 and number 10, next door neighbors together in a group. So I'll put some numbers here. I'll do number 9 on the left and number 10 on the right. And then remember that we've got this long one that was my controversy cranial nerve, number 11, that has all those branches that come off down here. This is the one, remember, that we say could potentially be considered a spinal nerve. So sometimes you'll see it called the spinal accessory nerve because it's down so low on the spinal cord. We have to go back up here to find number 12. So number 12 is actually up higher than number 11, number 12 right there. Okay, so cranial nerve number 8 did hearing and balance. Remind me again, what number did vision? What number is vision? Yeah, vision is cranial nerve number 2, the optic nerve. And then we talked about these, right? Eyeball movement, that was number 3, number 4, and number 6. Hey, here's my wink, wink, nudge, nudge. The cranial nerves that we just reviewed today, just now, we have talked about these cranial nerves in the cranial nerves packet and in the special senses packet. If it shows up in more than one lab packet, there's like a 95% chance it's going to show up on the lab exam. So we want to know all of our cranial nerves. Any of them are fair game, but please, please make sure we know these ones for sure. Because I could ask you about them when we're doing a question from the cranial nerves packet, or I could also ask you about them when we're doing a question related to special senses. It's like double dipping, right? If we learned it twice, we better make sure we really learned it because it's going to show up on the exam. So wink, wink, nudge, nudge to some of these cranial nerves. Make sure we really know those guys. The other thing that's showing up more than once are functions of our brain parts. So when we talk about special senses, cranial nerves collect this information and they send it back to different places in the brain. So let's remind ourselves about the places where this information goes. When we're talking about the process of hearing, which of my lobes in the brain helps me with the process of hearing? Who helps me with hearing? Yeah, exactly. The temporal lobe helps me with hearing. Here's a quick reminder for us that when we talk about the lobes of the brain, we are talking about parts of the cerebrum. That's the fancy name for the brain, the cerebrum, or the part of the brain with the lobes, excuse me. So in the cerebrum, we've got the temporal lobe that helps us out with hearing. Which of my lobes helps me with vision? Which lobe of the cerebrum helps me with vision? Yeah, exactly. Like Kira mentioned, the occipital lobe helps us to see things. When we talk about, let's see, when we talk about the frontal lobe, that's kind of a good segue for us. In general, when you talk about the frontal lobe, does that do sensory or motor? The frontal lobe. Yeah, so the frontal lobe is in front of the central sulcus, which means it's a part of the brain that does motor stuff. So if I had phrased my question here about eyeball movement a little bit more generically, if I just asked you for a lobe, we would say the frontal lobe. That's our motor lobe in the brain. But inside that frontal lobe, we have a special place that controls all of our skeletal muscle. So all of those muscles that you decide when they contract, there's one special place in your brain where they all live. That one special place in your brain where they all live is technically called the precentral gyrus, the bump in front of the central sulcus. We talked about, though, in lecture, how this is where we find that weird thing called the motor homunculus. The motor homunculus. So the map of all the places in the body that you control their movement of, there's part of it that helps us with moving the eyeballs. So controlling those muscles around the eyeball. So the answer I was looking for here with the part of the brain that helps us do eyeball movement, either the precentral gyrus or that motor homunculus. That's much more specific than just the frontal lobe here. When I talk about balance, we're actually not talking about a part of the cerebrum anymore. What part of the brain did we say was responsible for balance? Yeah, exactly. The part of the brain that does balance is the cerebellum. Remember, the cerebellum is easily influenced by alcohol. This is why when you've had a little too much to drink, you lose your balance and your coordination goes down, because that part of the brain would have been helping you with that, but it's under the influence of alcohol. So good review for us of the cranial nerves, of the functions of the cranial nerves, as well as the functions of some of those lobes of the brain. Before we move on, any questions? Let me check out my chat. So Kira asked a question about alcohol being lipid soluble. I know it's not a lipid. I'm not sure if it's lipid soluble or not. I think it actually is not. I think it's polar. But I'm not quite sure. I wonder if the reason why the cerebellum is so sensitive to it is because if you remember the choroid plexus, the blood vessels that leak out cerebrospinal fluid, I kind of suspect that it can leak out of those pretty readily, and there's some choroid plexus right next to the cerebellum. Yeah, so it could be, Kira, that it can cross the blood-brain barrier easily. It's pretty small, so I don't think that it is lipid soluble per se, but maybe it is. Somebody can prove me wrong with Google. Yeah, so Kaylin's saying it can diffuse across the blood-brain barrier. That would make sense, right? Yeah, so we're confirming. Let me go back to some of my other questions. Yeah, the process of vision is done in the occipital lobe, hearing done in the temporal lobe, and then our eyeball movement coming from the motor homunculus, so from the part of the brain that has a map of the whole body, which we find it in the precentral gyrus. Yeah, so we have a couple of questions we're thinking about from the exam. I gave some of you a question about a lesion that's on the motor homunculus. If you've got a lesion or a tumor, if you will, on your motor homunculus, in general, what might you predict would happen if you've got a problem with your motor homunculus? What might happen to you? Yeah, so the motor homunculus is all about movement, right? So if I've got a lesion that is found on the motor homunculus in the area around the eye, any of my eyeball movements, any of the movements of my eyelids, or basically anything around the eye, it is not going to do its movements. So if I've got a lesion on the motor homunculus, motor is always movement. If I've got a lesion on the sensory homunculus, what kind of information would the sensory homunculus be receiving? So does the sensory homunculus help us with vision? Yeah. Dr. Hollis is being tricky. The sensory homunculus only gives us general sensory information. So sensory, let's type this up here. Sensory homunculus. This is the thing in the post-central gyrus. This does general sensory info. So this is going to be touch, temperature. It's going to be things like pain, stretching. What it's not going to be, though, is vision. Because visual information, what you see, goes to the occipital lobe. What you see doesn't go to that sensory homunculus. So I can have an issue with feeling the temperature of something on my eyelids, or I can have an issue with not feeling pain if something were to touch my eyelid or touch my cornea. But for me to have issues with being able to see, that's an occipital lobe problem. That's not a sensory homunculus problem. Because remember, this only does general sensory information. The special senses are how we do vision. And we do vision in the occipital lobe of the brain. Yeah, so Kaylin's saying that she remembers that now from the notes. Yep, general sensory information. And we're going to talk more about general senses tomorrow in our lecture class. So consider that sensory homunculus is general sensory information. General sensory information goes through the spinal cord up to the brain. Special sensory information goes to specific lobes of the brain to be processed. Thumbs up, thumbs down, thumbs in the middle. What do we think? Perfect. Okay. Several thumbs up, and if you think of questions as we continue to work through, as always, feel free to pop them into the chat and we'll look at those. We're going to dive into hearing, actually hearing and balance, to be more precise. When we talk about the process of hearing and balance, we're talking about stuff that's going on in the ear. Now, the ear is divided into 3 regions. And you'll want to make sure, especially as you're working on the homework assignment and filling in your sheet, that we keep track of these 3 regions of the ear. So my first region of the ear that I've made a little bracket around right here, this is called the external ear, the external ear. This is the part of your ear that you can see, that you can touch. It ends with the structure right here that you can sometimes poke with a Q-tip if you're not careful enough. So this thing right here is called the tympanic membrane. The tympanic membrane. Let's see if I can type this without it looking crazy. The tympanic membrane. The job of the tympanic membrane is to divide the external ear from the middle ear. So we see the middle ear in this area. The tympanic membrane is just like a tympani, which is a really big drum. So the tympanic membrane, its easy person word is the eardrum. So the eardrum or the tympanic membrane divides the external ear from the middle ear. So the middle ear region, let's see if I can draw a little area around it. The middle ear region is what we kind of see here. The middle ear is a region that should be filled with air. That's its normal job. So the middle ear should be filled with air. Inside the middle ear region, we have some little bones called ossicles. There are 3 different ossicles. Let me write this word here. So middle ear bones, not bonds, the ossicles. So my middle ear ossicles, these are going to be, I'm going to color code, the malleus, the incus, and the stapes. Those are my ossicles, my middle ear bones. So the malleus is the one that is directly attached to the tympanic membrane, the malleus. The incus is right behind it, and it's kind of hard to see on this view. We'll see it better in a different view. The stapes is the one that's next to the internal ear. Next to the internal ear. So we really probably cannot see the stapes on this model. So the malleus is right here, the incus is right behind it. The stapes is going to be down lower. I'll show you on the other model. It's much easier to see. That is the end of the middle ear. The internal ear on this model is what we see right here. So these structures that we're looking at are part of the internal ear. The internal ear, put a little blue on them. The internal ear is made of kind of 3 main parts. One of those parts is the really important one called the cochlea. And the cochlea is what helps you to actually hear. It kind of looks like a snail shell. Try to say that like 5 times fast. Snail shell shaped cochlea. Snail shell shaped cochlea. It's very hard. So we've got the cochlea right here, shaped like a snail shell. Then you can see these little loops that are over here. Those are things called the semicircular canals. Semicircular canals. And we actually have 3 of these semicircular canals. They help us with balance in multiple different directions, which is why we have 3 of them. There's one other part, we don't ask you to label it, but we'll see it more later. This central part where those canals are connected to, we would call that part the vestibule. So you don't have to label it here. We'll label it on a different model. But the internal ear, primarily it's all this stuff that we can see back here. When we're inside of you, the internal ear is actually completely enclosed in bone. You wouldn't be able to see any of it because it's surrounded by bone. But this middle ear area inside of you, again, should be filled with air. Remember how I told you all my son has an ear infection? When you have an ear infection, there's all kinds of crud in what should be this air-filled space. So I told you how his tympanic membrane or his eardrum was swelling. It was pushing toward the outside because he's got so much stuff up here in his middle ear. So the middle ear is supposed to be empty. If you have an ear infection, not so much. The reason that kids get ear infections more often than adults is because of this thing right here. So this thing right here is called the pharyngotympanic tube. I'm not going to write that whole thing. I'm just going to draw a line. the pharyngotympanic tube. This is a tube that literally goes into your pharynx, your throat. Yeah, so it's also sometimes called the Eustachian tube. Absolutely. This tube is really helpful for you when you're on an airplane. When you feel a lot of pressure in your ears, it's because the pressure of the air outside is different from the air inside. So this tube allows you to swallow or chew some gum to equalize the pressure inside compared to outside. In kids, though, this tube isn't slanted downwards. In kids, this tube is a little bit more horizontal. So if they have anything draining out of their nose, it goes into this tube and it goes up into their ears. Where if you've got bacteria or viruses in that stuff and it's replicating, that's what leads to the swelling in the internal ear. So the pharyngotympanic tube in adults allows you to equalize pressure. In kids, it can be a source of ear infections. Yeah, so Kira asked if this was also shorter in kids. Yeah, absolutely. So in kids, it is horizontal and it's shorter. Lots of reasons why it's easier for bacteria and viruses to sneak up into this area here. So we've got the pharyngotympanic tube here. On the outside, on the other side of the tympanic membrane, we have this area called the external acoustic meatus. External acoustic meatus. This is something way back from bone markings in like lesson 6. We talked about how a meatus is a tube-like passageway or kind of a hole. So we've got our outside hearing hole, the external acoustic meatus, the place where those Q-tips go, right, when you're clearing out your ear. And then the part of your ear that you can see is this big part that we see over here called the auricle. So the auricle is the big part of your ear that is visible. This, yeah, or it's sometimes called the pinna, absolutely. So we're going to call it the auricle just for the sake of ease. Down at the bottom part, down here, is an area that we call the lobule. I like to remember the lobule as the part that you pierce typically. Put a little earring down here in the lobule. The auricle is the whole thing that kind of acts like a funnel to collect sound waves. Yeah, so Kira asked a good question. If we're not supposed to put Q-tips into our ears, what should we use? That's a great question. I'm not sure that doctors ever fully answer how you're supposed to clean your ears. I think the challenge with Q-tips in the ears is if you aren't careful, you can actually push any wax that's in there like back farther instead of getting it out. I know that they have special tools that use water to clean the ears, but I think most of us do not have one of those tools. So next time you see your doctor, you can ask, what am I actually supposed to do to deal with my ears? I had seen a question earlier about which part of the ear we would consider the tympanic membrane part of. I would tend to agree that it's probably not part of either the external ear or the middle ear. It's the dividing line between the two of them. So when you're making your list in the packet, you can put it on both lists if you want to, like highlight it though, just so you remember that this is my dividing line that divides up the external ear from the middle ear. And then there's a thing we'll look at in a moment in the middle ear called the oval window. That's the same deal like the tympanic membrane. It divides up the middle ear from the internal ear. So just make sure we know those dividing lines. You don't have to group them middle ear or external ear. They're literally the gate between the two. All of our anatomy on this model is the same, so I'm not going to cover everything. What I am going to cover, though, are those ossicles. So I want to show you a little bit better because we can see them more clearly now. The ossicles are those little bones that were inside the middle ear region. So we have the middle ear, remember that's air-filled. There's that external acoustic meatus down below. When we see our ossicles, the one that is attached to the tympanic membrane is the malleus. And when you're trying to remember that, this word malleus looks like a mallet, which is something you can use to play a drum. So the tympanic membrane is the eardrum. The malleus is the mallet or the bone that's attached directly to it. Next door neighbor to the malleus is the bone called the incus. So we can see the incus next to it. And then the last bone that you see here in gray is the one called the stapes. So this word stapes literally means stirrup, kind of like that thing you would see on a horse's saddle where you put your feet. So the stapes is the last part of the middle ear. It's right next to that thing I mentioned called the oval window. So this is the last structure of the middle ear, the stapes. You can see those semicircular canals again really well on this model. You can also see that cochlea really well here as well. Our next model, imagine that we took this, only this part here, and we zoomed in on it. That's what we're looking at here with this model of the inner ear or the internal ear. So some of the parts that we've already talked about, you can see that snail shell shaped cochlea. So I can see it from the front and from the back, the cochlea. You can see, by the way, when we're in the back view, this right here is what we call the cochlear branch of the vestibulocochlear nerve. This little thing right here is the vestibular branch of the vestibulocochlear nerve. Now, we're not going to be that specific and ask you to name the two branches, but when we circle these things together, so when you see this, just know that the reason it looks like it's two parts is because it actually is. This is the part that does hearing. This is the part that does balance. So this is the vestibulocochlear nerve coming off of the cochlea over here, or the cochlea right here, and coming off of the vestibule, which is this area over here. You can see those semicircular canals again when we look at this model. So there's three of them. We can see those three. What we can see better on this model, though, is a part of those semicircular canals. It's got a little dot on it on our models, and you can kind of see it here over here as well. So that is a part of the semicircular canals called the ampulla. So here I'll put a circle. Each semicircular canal has an ampulla, and when we talk about the way that your body actually does balance, it uses the ampulla. That's where we actually collect balance information, is in that part of the semicircular canal. So each of my semicircular canals has this area where it's wider. That place where it's wider is called the ampulla. That's where we actually do balance information. We also have inside this big area in here, we have two fluid-filled structures. So now we're inside the area we might call the vestibule. We have a structure that's up higher called the utricle. I like to remember the utricle is up. So they will start with U. Utricle, and then the larger saccule that's down below it. So the utricle and the saccule, these are going to help you out with balance, like the kind of balance when you get in an elevator and it goes up and you feel that initial jolt. That's because of the utricle and the saccule. When you're spinning in your desk chair, that's because of these semicircular canals. So all of these things help us with balance. They're feeding into that vestibular part. This over here helps us with hearing. They're feeding into the cochlea. Thumbs up so far with this, or any questions before I move on to my next model? Okay, so to help us get ready for the next model, I want to show you a picture. You have this in the guided lesson, correct? Is this picture in your guided lesson, hopefully, for lab? Probably, Kira thinks. Yeah, so if not, I know it's in the lecture guided lesson too. So you have this picture somewhere. This picture is excellent because it shows you how the process of hearing works. So hearing starts with sounds that come in through that external acoustic meatus. They vibrate against the tympanic membrane, which makes this malleus bone right here vibrate back and forth. which makes the incus bone that's attached to it vibrate back and forth, which will make ultimately the stapes vibrate back and forth. At that point, we bump into a structure called the oval window, which is my dividing line between the middle ear and the internal ear. Where you actually do hearing is inside the internal ear where the cochlea is. Now, this picture looks pretty weird because we haven't seen anything like this. But imagine that I took that snail shell that was all curled up and I laid it a little bit flat. So the snail shell is actually made out of 3 different tubes of fluid. So these 3 different tubes of fluid get the sound waves at different times. So sound waves first go into a tube that's called Scala vestibuli. So when we're first hearing a sound, Scala vestibuli is the first fluid-filled tube that the sound waves go to. These sound waves are going to bounce and bounce and bounce until they find the specific part of your ear where you can hear them. So we hear high-pitched sounds at a different place than we hear low-pitched sounds. Sounds are going to bounce around and bounce around until they find the place that they're heard. When they find that place, those sound waves actually transfer into our middle fluid-filled tube, which is called the cochlear duct. So the cochlear duct is the middle fluid-filled tube where sound waves are actually perceived, where we actually hear them. Once you've heard them, though, we don't want to keep hearing them. We want to hear them once, and we want to get rid of them. So we hear them in the cochlear duct, and then we send them to this second part right here called Scala Tympani. You can see its name right here, Scala Tympani. This is the third fluid-filled tube. Scala Tympani takes those sounds out and actually sends them back to the middle ear. I like to say that this place they let them go, called the round window, this is kind of like a garbage disposal. So this is where sounds go to die. When you've heard them, they first get into Scala vestibuli. They find the place you can hear them, so they go into the cochlear duct. And when you've heard them, we spit them out into Scala Tympani, all the way out to the round window where they spit back out here. There's nothing to catch them. They go away. The 3 fluid-filled tubes of hearing. It's important for us to know the order that they go in. So we start with Scala vestibuli, then we go to the cochlear duct, then we go to Scala Tympani. I'm pretty sure we put this note in your lab packet, but if we did not, please write down for yourself. A really good way to study this is to make a flow chart. Remember how we did those flow charts with muscle contraction? When you're studying the process of hearing, a flow chart is invaluable. You really want to be able to build a flow chart that shows you the order things go in. So from the external acoustic meatus, through each of those ossicles, through each of these fluid-filled tubes, if you can build yourself a flow chart like that, that will help you immensely to remember the order things go in. Yeah, so Temiseo says there's a note about that in the study guide. Definitely make sure to do that. It will be a huge help for you as you're studying. Okay, we've talked through what hearing looks like on this picture. Now I need you to use your visualization skills with me here. Imagine that I cut a slice right here. So I chopped my cochlea right here, and then I kind of turned it around. So right now it's laying flat on your screen. Let's imagine that I turned it so that you're looking down the middle of it. So I took this cochlea, I made a slice right here, and I rotated it. This is what you're going to see. So we've got 3 fluid-filled tubes. Now instead of seeing them next to each other, I'm staring straight into these tubes. These are the exact same tubes that we just talked about, but in a different view. So let's go through and label our cochlea model here, where we can see our 3 fluid-filled tubes and some of the other important structures. So we'll start up here. This top tube up here is where sound waves go when we first are trying to hear them. When I'm first trying to hear sound waves, was it my Scala Tympani or my Scala vestibuli where I first sense sound waves. Do we remember? Which of those scalas? Yeah, exactly. Scala vestibuli is the first tube that I send my fluids into, or I send my sound waves into. This fluid-filled tube, those sound waves will bounce and bounce and bounce until they find the place on my middle tube, which is called the cochlear duct, until they find the place where they can be heard. So we'll bounce and bounce and bounce and bounce until we find the place in the cochlear duct where we can be heard. Then we'll start to bounce around in the cochlear duct. We will get heard, and then we'll bounce into Scala Tympani down below. So Scala Tympani was my other fluid-filled tube. Remember, this is my recycling tube where I send those sound waves out to be dumped in the garbage disposal. So on the top, we've got Scala vestibuli. In the middle, we have the cochlear duct, and down below, we have Scala tympani. I like that. Kira said tympani kind of like trash. That's perfect. So the tympani part is the trash part. Time to take out the trash, get rid of those sound waves. Absolutely. When we're doing the process of hearing, what actually does the hearing are these special little cells that we see right here. These special little cells are cells called hair cells. Hair cells. Now, here's a wink, wink, nudge, nudge for lesson number 13 in lecture. Hair cells are involved all over the place in special senses. We use them for every kind of special sense except for vision. So hearing, balance, taste, smell, hair cells all over the place. These little cells right here are called hair cells, and they get their name because literally they've got little hairs on top. So their dendrites are arranged in such a way that they kind of look like little hairs in there. So there's my little hairs on top of the hair cells. So the way that the process of hearing works is we actually push on these hairs, we change their shape to be able to hear messages. So here's how it works. I've got these hair cells. These hair cells are sitting on top of something called the basilar membrane. The basilar membrane, let me label it. The basilar membrane is the basement that these cells are sitting on. So sound waves are bouncing, bouncing, bouncing. We've found the place where they can be heard, so they start bouncing inside here. Well, what happens when the sound waves bounce inside here is the basilar membrane starts to bounce up and down as well. And these little hair cells are sitting on top of it, so the hair cells start bouncing up and down too. The only problem is this thing right here that's right on top of the hairs, that's forming kind of a tent on the hairs, this does not move. So these cells are bouncing and bouncing and bouncing. This doesn't move. Those hair cells start to bump into it. So this thing up here is called the tectorial membrane, and this thing doesn't move. So it's chilling out here. When these hair cells start bouncing, they're going to press against it, and that's going to register as a sound. We perceive that as a sound. So my sound wave bounce in here, they make the basilar membrane bounce up and down. We bend those hairs. But once we're done, once we've made this basilar membrane bounce up and down, we'll actually send those bouncing sound waves down here into Scala tympani. So when you stop hearing, it's because the basilar membrane stops moving and those hair cells don't keep bumping into that membrane anymore. When we talk about how you hear, big picture, we call what we hear with the spiral organ. So the spiral organ is kind of all of this stuff right here. So if you see a bracket when you're working on your homework assignment, organ, the spiral organ. Yeah, our Elapine is right. The technical name for it is the organ of Corti. we like spiral organ because that's easier to say, right? So the spiral organ is my tectorial membrane, my basilar membrane, those hair cells. Essentially, it's all the stuff that I use to actively hear. That's the spiral organ. So on this model, again, remember, we took that picture on the last slide and we rotated it around to see the 3 fluid-filled tubes that sound waves go into, and to help us see those cells that help us do hearing because they bump into the membrane that doesn't move. We have another model that shows this. This model doesn't show me all 3 of the tubes together, like fully made. So let me show you. You can always add to your picture these kinds of circles like this. Then we can see our 3 tubes. So up here at the top is where we would have scala vestibuli, where sounds first go. Then we've got our cochlear duct here in the middle. And then down below it is scala tympani in the bottom. You can see again when we're looking at this model, here's those little hair cells. You can see them here. Their hairs aren't quite as pretty, although these hairs look a lot more like baby hairs. Like my son has really fine hair. So we see really fine little hairs on top of these hair cells here. Then we've got that tectorial membrane that doesn't move, the thing that these hair cells bump up against. The basilar membrane that they're sitting on top of, and all of this that we're looking at here being our spiral organ. I believe that is our last hearing structure. Any last minute questions about hearing? Check this out. We got ear emojis. All right, give me a thumbs up or some other emoji if you're feeling good. I was going to say, if we stayed quiet for too long, I was going to think that I was all alone, and that would make me sad. Yeah, Kara said that our hearing is not great. I feel you. It's all good. All right. Well, remind me before we move on, which cranial nerve was the cochlea sending information to? What cranial nerve number are we talking about here? Yeah, cranial nerve number 8. So the cochlea sends all of its information to the vestibulocochlear nerve, cranial nerve number 8, the process of hearing. Absolutely. Let's go to the process of vision, the process of seeing. Now, full disclosure, on this model, what is going to be the trickiest and also possibly the most important for you to figure out is the location of each of the different muscles that attach to the eyeball. We have 2 different eyeball models. You've got all kinds of pictures of them showing you those models in all kinds of different views. I'm going to try to give you some hints to help you figure out where everybody lives to make sure we stay on track. When we're talking about the eyeball in general, let's start with some general terms here. This black part in the very middle of your eye, this is actually the opening in your eye. So this is the pupil, the open space in the middle of the eyeball. Surrounding the pupil is the colored part of your eye called the iris. Hey, fun fact about the iris, the same pigment that gives you your skin color gives you your iris color. What pigment gives us the color in our skin? Yeah, exactly. Melanin. So it's the same melanin that gives you your skin color, but also gives you your eye color. This model shows a blue iris. We also have people with green eyes and brown eyes, hazel eyes. Do you think that blue eyes have a lot or a little melanin based on some of the other colors that are out there. Yeah, Kira's right. Blue eyes are where we have the least melanin of all. Blue and like really light green, very little melanin. Brown, especially dark brown, lots of melanin. So the same kind of melanin that gives you your skin color gives you your iris color as well. Outside of the iris, we have the white part of your eye, or let's be honest, the part that's supposed to be white if you're sleeping enough. That's the part of your eye known as your sclera. So the sclera is the outer white part of the eye. The sclera is really fibrous and tough. It needs to be strong so that all of these different muscles that we're going to be learning can attach to it. So the sclera is here on the outside. What we don't have on this model that you have in the very front of your iris and your pupil is the clear layer called your cornea. So we'll draw our cornea on in a different view, but that's the very front part of your eye that's in front of these structures that we see here. This particular eyeball model that we're looking at here shows us several of the bones of the skull. And the reason we have these bones of the skull is to help us to identify lateral versus medial. So this bone right here is where a pair of glasses would sit on. Which bone do glasses sit on your face? What's the bone that we have right there? Yeah, exactly. Okay, so this is my nasal bone. Next to the nasal bone and underneath the eye, what's the bone that we have right here? It actually extends down to your jaw when we're looking at it. Yeah, exactly. So this one right here is the maxilla. The one right here is one of those bones that helps to make your cheekbone. This one over here is the zygomatic bone. Absolutely, the zygomatic bone. And then up above the nasal bone, we've got your big frontal bone. Your big frontal bone. Okay, this is a great review of the bones of the skull. Another thing that we're going to find on that lab exam, the bones of the skull. We want to know where these bones are on this model because it's going to help me tell lateral from medial. Is it the nasal bone or the zygomatic bone that we would say is medial on this model? Zygomatic or nasal? Which one is medial? Yeah, exactly. The nasal bone, your nose, is more toward the middle than your zygomatic bones, the ones that make up your cheeks. When you are looking at this model head on, so we're staring into the pupil of this eyeball, this is the medial side of the model. This is the lateral side of the model. That matters because when we're looking at the names of eyeball muscles, we've got some that say lateral and medial. So we've got to know, step one, that this is the medial side and this is the lateral side. We know that because of the nasal bone. That tells me where the middle is. The name of these muscles has the word rectus in them. If you remember back from our leg muscle stuff or from our abdominal muscle stuff, rectus means that we're parallel. So rectus abdominis is the six-pack muscle. Rectus femoris follows your femur. Rectus means parallel. When we're talking about eyeball muscles, the rectus muscles are parallel to the optic nerve, which is something we can't see it in this picture, but imagine straight behind the pupil, sticking straight out and back into the skull. That's the way the optic nerve is going to work. So any of my muscles that you see from the front here that are just kind of like a block because they go straight back, all of those are rectus muscles. So we said that the nasal bone is medial. That makes this one right here the medial rectus muscle. The medial rectus muscle. And that makes the one on the other side of the eyeball the lateral rectus muscle, the lateral rectus muscle. When we go to the top and the bottom of the eyeball, we actually have two different kinds of muscles there. We've got these rectus muscles that we talked about before that go straight back, but then we also have these muscles that you can see here and here that go at an angle. When we go at an angle, remember from the abs that we call that oblique muscles. So when I'm talking about the top of the eyeball, would I use the word superior or inferior to describe these two muscles up here? Are they superior or inferior? Yeah, exactly. These are my superiors. So we've got the superior rectus right there and the superior oblique next door. Superior oblique, superior rectus. So then we go down below. These are going to be my inferior muscles, right? These are my inferior. So we've got the inferior rectus and the inferior oblique. 100% what I think is going to be the hardest about this model is figuring out the muscles, but I promise it's not as hard as you think it is if you can just figure out where the middle is and where the top or the bottom is. Because all of these muscles that we see on our list here to know about from the eyeball, all of these muscles have directional terms in them. All of them have stuff that tells us what direction the fibers are going. Are they going straight or are they going out? So use all of those clues that you can to help you figure out where these different eyeball muscles are. When we start looking at other views of this model, it's important for us still to figure out lateral versus medial. Now, I'm pretty sure in your packet, do we have a label underneath this picture? I'm pretty sure we do, right? Did I tell you what view we're looking at? I did. Yeah. So this is us looking at the lateral view of this model. I told you point blank that it's the lateral view. If I hadn't have told you that, the fact that over here is that frontal bone still. That frontal bone that we labeled that helped us find the medial part. Here's my frontal bone up here that's sticking out. My zygomatic bone, my cheekbone, is right here. So this is kind of like if somebody went next to you and they stared into your skull. You're looking forward, they're staring at you from the side. That's the view we're looking at here. So that means, and notice you can see here really well how this is a straight line muscle. That means that this one right here is my lateral rectus, muscle because it's on the outside. We didn't ask you to label it, but see this muscle that's kind of back here, almost hidden? That's the medial rectus that's on the other side of the eyeball. The superior rectus is up here. We don't see it extending all the way back, but it would go back just like that. And the inferior rectus started on the bottom and is extending up right here. So superior and inferior, lateral and medial. This is actually part of that oblique muscle. So you can see it kind of attaching at an angle up here, and then it kind of angles backwards. So this is my superior oblique muscle that I can see up here. I'm checking out our things we have to label as well. We have to label the optic nerve in this view. And when we're looking at our eyeball in this view, see this large white structure right here? We'll see it better in another view. It extends all the way into the eyeball. This is that optic nerve. So the optic nerve, cranial nerve number 2, I can see it right here. We ask you to label the optic canal, which is one of your bone markings from the skull chapter. So the optic canal, we can kind of, it's not fully intact right here, but imagine that there's a ring of bone. So if all of this was bone tissue, which it is when you're alive, the optic canal is the open space where the optic nerve is inside of. So the optic canal, you can draw yourself a little circle to show you that it's an opening in the bone tissue. That's the optic canal, the space where we'd find it. I got multiple emailed questions about this view of the model. So I want to give you a hint when we're looking at this view of the model. It's labeled for you that this is the superior view, meaning we are looking from the top down at our muscles here. To help us figure out lateral versus medial when we're looking from the top down. So if someone's standing over your head, looking into your mind, or looking into your eyes, I guess, through your mind's gone. Here's the structure right here that is most important to help us figure out lateral versus medial. Does anyone know either what bone or what bone marking we're looking at right here? What is this? Yeah, so Kira's helping us out here. This is something called the ethmoid bone. The ethmoid bone. We did not technically ask you to label it on this picture, but it is a bone you learned in the skull. The way we can identify the ethmoid bone are all of these little spots. These little spots right here are called the cribriform foramina. The cribriform foramina. That is a bone marking. Make sure we go back and review that one. The cribriform foramina are literally little holes that I have in the ethmoid bone. These little holes go down into the nasal cavity to allow you to smell. So the olfactory nerve sends on little shoots or little, they're almost kind of like octopus tentacles, down through these little holes into the nasal cavity. So, long story short, these are on top of the nasal cavity. Does that mean that this or this, so we'll say number 1 or number 2? Who is medial? Number 1 or number 2? If my nasal cavity is right here. Yeah, exactly. Exactly. This is medial. This is medial, which means this is lateral out here. So when you're labeling your muscles, in particular the medial and lateral rectus, I guess maybe we didn't ask you for lateral rectus. When we're labeling these things, medial is here because my nasal cavity, my nose, is down below here. This is out with my zygomatic bone, way here on the outside. So we've got our eyeball. Notice, by the way, look how clearly we can see that optic nerve. So there's that optic nerve coming out of the eyeball. We've got the optic nerve coming out of the eyeball. We've got the lateral rectus muscle that's out here on the side. The medial rectus muscle that's here in the middle. You can see your inferior rectus muscle underneath the optic nerve. So there's the inferior rectus muscle. And the superior rectus muscle up here. We could draw on, by the way, if it was still there, that muscle would go back like this. The superior rectus muscle. So I'll add some red for it. There's the superior rectus for us. We've also got that superior oblique that we can see up here. So this is your tip-off right here. We've got to be able to identify those cribriform foramina, the little tiny holes in the ethmoid bone, because that tells us where medial is. That tells us where the middle of our model is. So we know that this is the lateral rectus and the medial rectus is down here. As confusing as the bone model can be, it is important, or it's helpful for us because it shows us medial versus lateral. When we look at this eyeball model, we don't have bones to tell us that anymore. So there's actually a couple of different ways that we can tell where lateral versus medial is when we don't have the bones anymore. One of them, probably the easiest to use, is this one right here. For my friends who had a chance to work on this lab, what are we looking at right here? Do we know what this thing is? Yeah, exactly. This thing right here is the lacrimal glands, the lacrimal gland. So the lacrimal gland helps you to cry, helps you to make tears. Both of your eyes have a lacrimal gland. It is found superior, so here's the top part of my eyeball, and it is found lateral. So I know you have this question in your lab packet. What helps us to find the lateral side of the eyeball? It's the lacrimal gland. The lacrimal gland is always on the lateral aspect of the eyeball. I'll mention, though, that the other thing that can help us tell the difference between lateral and medial is the direction that our superior oblique muscle goes. The superior oblique muscle always goes toward the medial side. It attaches toward the middle. So I've actually got two tip-offs when I'm looking at this model. The lacrimal gland, very easy to see on the lateral side, but also that oblique muscle going toward the medial area. Notice we put some little stars here for you. Our muscles, especially those rectus muscles, didn't show up very good on our picture here. So we need to add some rectus muscles. Here's the first rectus muscle that I just added. Which rectus did I just add? What's the name of my rectus? This one right here. That first rectus. Yeah, exactly. I'm on the top. So that's superior rectus on the top. Hey, now I'm going to draw the rectus that's on the side with the superior oblique, the way it's going toward. Who's this guy? Yeah, this one would be medial. Medial because it's on the same side as that oblique is going to. And then I can add for myself my lateral rectus as well. The inferior rectus we can actually see. That's still down here, the inferior rectus. So again, I'm looking at this model. Notice that I've got my lacrimal gland right here. Since I have my lacrimal gland right here, would I say that this is the medial or the lateral rectus? Who's this, since I see that lacrimal gland? Yeah, exactly. This one is my lateral rectus because it's on the same side as the lacrimal gland. The lacrimal gland on the lateral side, lateral rectus right here. And I can tell when I'm looking at the model here, here's that superior oblique muscle going toward the medial side of the eyeball. So this is my medial rectus that I see here. Medial rectus on the same side as that superior oblique is going to. If we look inside our eyeball model, we can start to see some of the different layers that make up the eye. So there's a couple of things we should add to this picture. One of the things we should add to this picture is that clear layer that would be in the front of your eyeball. What's the name of the clear layer in the front of your eyeball? The part where contact lenses sit on top of? Yeah, the cornea, absolutely. So I just drew on my cornea here to remind myself that the iris is not the first thing in my eyeball. I got the cornea out here. See how there's this nice little indentation right here, this nice open space? That is where it asks you to draw where the lens would be. That is where it would be in the space right here. Oops. So I'm going to draw a little lens right here. The lens actually rests inside. This that I see right here is something called the ciliary body. It's a group of muscles that pull on this lens. They expand it and contract it to help you be able to see things up close. Yeah, so Kaylin's mentioning that she had some issues with her cornea getting damaged in a car accident. If you all happen to remember when we learned about the little girl Gabby who didn't feel pain? Yeah, exactly. Remember that the way that they figured out that she couldn't feel pain is that she literally scratched her cornea so much that like it was damaged. Like it was white because she had damaged it so badly. So your cornea, we're going to talk about in lecture tomorrow, has a bunch of sensory neurons in it that if anything touches your cornea, it's supposed to make you blink. Well, think about Gabby. She was scratching her cornea and it didn't make her blink. So it was pretty intense. Yeah, so okay, yeah, so Kaylin said luckily she didn't have to have surgery for her cornea getting torn, but no more contacts and have to the antibiotics and the steroids. Yeah, absolutely. Cornea is a very important part of the body to keep healthy. That's for sure. There's fluid that we find up here. Something you actually are labeling this in your packet. So here we'll say it while we're at it. The fluid that I find right here by the cornea is something called aqueous humor. So aqueous humor is really watery between the cornea and the lens. And in the back, inside most of the eyeball, we have what's called vitreous humor. And vitreous humor is a lot more kind of like jello, if you will, lots of proteins that are back here. So we've got this cornea protective layer that keeps the aqueous humor in place, unless, like Kaylin was talking about, unless some glass shatters through it and causes some issues. So that is crazy, Kaylin. I'm so glad that you're able to see still. I do know that corneas are something that can actually be transplanted from various people. They are immunoprivileged, so you're not likely to reject a donor cornea, interestingly. So I'm glad you didn't need one, Kaylin. But this part of the eyeball can actually be donated. We can trade that amongst people. So I'm reading Tien's question. So it can heal itself, Tien. The blood supply to the cornea is not nearly as good as the blood supply that we see back here in the parts of the eyeball where we see. So it's not that it can't heal itself, it's just that it can't heal it very quickly. It's kind of like how if you hurt a muscle, muscles have a lot more blood supply than a tendon or a ligament. So if you rip a tendon or a ligament, those take a really long time to heal, or they might not heal because there's so little blood supply to those tissues. So it's kind of the same deal with the cornea. There's not nearly as much blood supply to the cornea to help it heal as much. Aqueous humor can help because that has oxygen and some nutrients in it, but they don't have all the blood supply. So see this pink part of the eyeball back here? This pink part right here is like all blood vessels. So this pink part here inside the eyeball is a layer that we call the choroid layer. So my pink part of the eyeball, choroid, choroid, we'll put a little line on there. That's the blood supply of the eyeball. So blood supply layer. Lots and lots of blood vessels that are helping to keep this inner yellow layer alive. This inner yellow layer, called the retina, that's where you actually see the retina. You can actually have your retina become detached from the choroid layer. So if the retina detaches from the choroid, you don't have very much time at all before the retina starts to die. The retina needs constant blood supply. So that's why it's literally sitting on top of all these blood vessels to have the blood supply that it needs. You can see that the outer part, so on the outside of the choroid layer, you can see that white layer on the outside of the eyeball. That was the sclera, the white outermost layer of the eyeball. Really thick and really tough. There is a spot in the retina. I'm going to circle it here for us. This spot in the retina back here, this is something called the Macula Ludia. The Macula Ludia. This is the place where when you see something, so light passes through your pupil, your lens focuses it. It's trying to focus it right here on a part of the eyeball called the Macula Ludia. The Macula Ludia is where you see the clearest. So this is where light is supposed to be focused inside your eyeball. the Macula Ludia. So when your vision is working correctly, that's where the light's going. We will talk about in lecture next week if you're nearsighted or if you're farsighted, if you have astigmatism. All of those are issues with the light not focusing on the Macula Ludia. So we can't see clearly if the light's not focusing here. Yeah, so Temiteo made a great connection when we talk about macular degeneration, which is a way that someone can go blind. That is an issue, absolutely, with our neurons that live here in the Macula Ludia. So some of those rods and cones that live here. Actually, it's just cones that live here, that specific type of cell. They see very clearly. Lots of them here. If they start to die, that can be an issue with us. Next door to the Macula Ludia, by the way, where we see the clearest, we also have this area right here where you can see those blood vessels going into the eyeball. We call this area right here where the blood vessels go into the eyeball, we call that the optic disc. The optic disc is the blind spot in the eye. So the reason that this is the blind spot is because there's literally no seeing cells here. There's no rods and cones here because this is where blood vessels go into the eyeball. It's also where all of those neurons are leaving the eyeball to form that optic nerve here in the back. Any questions about this model before we look at the last one? Edith's good. Kira's good. Sending us an eyeball just because I can. All right, well, the one last model that you're studying for class this week, imagine that we took a little section right here of inside the eyeball and we zoomed in really close on these layers here. So this model shows me up close and personal what my different layers are like. So we're going to start here at the bottom of the model, which shows us the outermost layer of the eyeball. What was the name of the outermost layer of the eyeball, that whites of your eyes? Yeah, exactly. So this outermost layer, these white stuff that we can see down here, that's the sclera, the white part of your eye. Notice right on top of the sclera, you can see this area that has all of those blood vessels in it. Look at these huge arteries and veins in here. What was the name of the layer that was the blood supply for the eyeball? Because here's that blood supply layer that we're looking at here. Yeah, exactly. This is my choroid layer, So then we don't have you label it as a group, but I'll put a group label on it here. Then all this stuff we're seeing on top of it is the retina. So the retina, the part of your eyeball you actually see with, is made out of 3 different kinds of cells. And when we talk about the 3 different kinds of cells in the retina, a lot of times we like to say that the retina works backwards. So what I mean by that is light comes into your eyeball up here. Let me get an arrow here. So the way that light travels is, pencil would be helpful, is like this. Light comes from the outside. It passes through these outer cells, until it can be perceived by these cells down here. So these cells that we see down here are cells that we call rods and cones. And I promise you on the lab exam, I'm not going to ask you to tell the difference between a rod and a cone. So this layer right here, these are my rods and cones. And these are the cells that actually detect light. The rods and the cones talk to this next layer of cells, put in a box around here. This next layer of cells are what we call the bipolar cells. So bipolar cells, remember that bipolar neurons had dendrites where they received information. They sent that to the cell body, and then they had an axon that sent it away. Bipolar means 2 extensions. So then we send it to those bipolar cells. So first the rods and cones see it. Then they tell it to the bipolar cells. The bipolar cells summarize it a little bit, and they send that information to the group of cells we see here at the top. These cells up here at the top are called the ganglion cells. Ganglion cells. These are the cells that are actually going to form the optic nerve, the cells that actually leave the eyeball. So the ganglion cells, they're the first ones that light bumps into, but they don't listen to the light. They don't care. That light also bumps into the bipolar cells. They don't care. The only cells that care about light are the rods and the cones that we see down here at the bottom of the retina. So make sure we can identify our layers of the eyeball, and make sure we can identify our cell types when we're looking at this model. Yeah, so Kaylin, light's coming into the eyeball. It passes through the ganglion cells at the top. They don't care. They can't respond to light. It passes through the bipolar cells. They can't respond to light. It hits the rods and cones. Rods and cones, literally, they have a protein inside of them that changes shape so that they can respond to light. So the rods and cones are excited. They talk to the bipolar cells. They tell the bipolar cells what they're seeing. The bipolar cells summarize it, and they tell that information to the ganglion cells. And the ganglion cells summarize that information and send it out of the eye using that optic nerve. So the ganglion cells leave the retina. They go out through, here we're going to go back. They go out through the optic disc. This is all those ganglion cells. They're axons leaving the retina, going back to the brain. With that, we are through our last lab packet. Woohoo! Any last minute eyeball questions or ear questions before we call it a day? I owe you all a penguin. We don't get them nearly often enough. Got a hiding monkey. I love it. My penguin is using a magnifying glass to see things. Or he's eating a lollipop. It looks about the same. All right, well, friendly reminder that you have 2 visible body assignments due by Friday of this week. Please, please take the time to do that anatomy search when you're working on especially stuff in the eye, but the ear too. Do that anatomy search in the practice assignment so you can see where visible body thinks each of these parts are. Do that before you do the assignments. That's Friday night. And remember, Sunday night is when you need to complete those 2 Blackboard assignments about special senses as well. So good luck on those assignments. Remember that tomorrow's lecture, we're going to work on the spinal cord. So if you have a chance, try to start working through that material from the lecture lesson on the spinal cord. We will plan to do that tomorrow, and we'll just keep trucking ahead. So have a great afternoon. I hope to see you tomorrow.